Everyone, welcome to the video lecture for Unit 4, Module 8.1. As always, here are your learning objectives, so be sure to reference these when studying for your lecture quizzes as well as the unit exam. Let's go ahead and get started. So the urinary system removes most physiological waste. In this module, we're going to consider the functional organization of the urinary system and describe how the kidneys move metabolic waste from the circulation to produce urine. We'll also explain the major regulatory mechanism controlling urine production and concentration, and identify how urine is transported to the urinary bladder and released by the body through the urinary tract passageways. The urinary system has three major functions. 1. Excretion, which is the removal of organic waste products from body fluids. 2. Elimination, which is the discharge of these waste products into the environment. And 3. Homeostatic regulation of the volume and solute concentration of the blood. The organs of the urinary system include the paired kidneys, the paired uterus, urinary bladder, and the urethra. The two kidneys perform the excretory functions of the urinary system. These organs produce urine, which is a fluid that contains water, ions, and small soluble compounds. Urine leaving the kidneys flows to the urinary tract, which consists of paired tubes called ureters. To the urinary bladder, a muscular sac for temporary storage of urine. After leaving the urinary bladder, urine passes through the urethra, which conducts a urine to the exterior. The urinary bladder and the urethra eliminate urine. This process is called urination or micturition. In this process, the muscular urinary bladder contracts and forces urine through the urethra and out of the body. The urinary system removes waste generated by cells throughout the body, but it has several other essential homeostatic functions. They include the following: regulating blood volume and blood pressure by adjusting the volume of water lost in the urine, releasing erythropoietin, and releasing renin. Regulating plasma concentrations of sodium, potassium, chloride, and other ions by influencing the quantities lost in the urine. The kidneys also control calcium ion levels through the synthesis of calcitriol, helping to stabilize blood pH by controlling the loss of hydrogen ions and bicarbonate ions in urine, conserving valuable nutrients by preventing their loss in urine, which removes organic waste, especially nitrogenous waste such as urea and uric acid, assisting the liver in detoxifying poisons and during starvation, deaminating amino acids for their metabolic use by other tissues. And these activities are carefully regulated to keep the composition of blood within acceptable limits. A disruption of any one of them has immediate consequences and can be fatal. The kidneys are located on either side of the vertebral column, between vertebrae T12 and L3. The left kidney lies slightly superior to the right kidney. The right kidney is slightly inferior to the position of the right lobe of the liver. The superior surface of each kidney is capped by an adrenal gland. The kidneys and adrenal glands lie between the muscles of the posterior body wall and the parietal peritoneum in a retroperitoneal position. The kidneys are held in position within the abdominal cavity by 1. The overlying peritoneum, 2. Contact with the adjacent visceral organs, and 3. Supporting connective tissues. 3 concentric layers of connective tissues protect and stabilize each kidney. They are 1. The fibrous capsule, which is a layer of collagen fibers that covers the outer surface of the entire organ. 2. The perinephritic fat, which is a thick layer of adipose tissue that surrounds the fibrous capsule. And 3. The renal fascia, which is a dense fibrous outer layer that anchors the kidneys to surrounding structures. Collagen fibers extend outward from the fibrous capsule through the perinephritic fat due to this layer. Posteriorly, the renal fascia fuses with the deep fascia surrounding the muscles of the body wall. Anteriorly, the renal fascia forms a thick layer that fuses with the peritoneum. In effect, each kidney hangs suspended by collagen fibers from the renal fascia and is packed with a soft cushion of adipose tissue. This arrangement prevents the jolts and shocks of day-to-day living from disturbing normal kidney function. If the suspensory fibers break or become detached, a slight bump or hit can displace the kidney and stress the attached vessels and ureter. This condition is called a floating kidney. It may cause pain or other problems from the distortion of the ureter or the blood vessels during the movement. A typical adult kidney is reddish-brown and about 10 centimeters long, about 5.5 centimeters wide, and about 3 centimeters thick. Each one weighs about 150 grams. The ileum, which is a prominent medial indentation, is the point of entry for the renal artery and the renal nerves. The ileum is also the point of exit for the renal vein and the ureter. The fibrous capsule covering the outer surface of the kidney also lines the renal sinus, which is an internal cavity within the kidney. The fibrous capsule is bound to the outer surfaces of the structures within the renal sinus. In this way, it stabilizes the position of the ureter, renal blood vessels, and the nerves. The kidney itself has an outer cortex and an inner medulla. The renal cortex is the superficial portion of the kidney in contact with the fibrous capsule. The cortex is reddish-brown and granular. The renal medulla consists of 6 to 8 distinct triangular structures called renal pyramids. The base of each pyramid touches the cortex. The tip of each pyramid, which is a region known as the renal papilla, projects into the renal sinus. Each pyramid has a series of fine grooves that converge at the papilla. Bands of cortical tissue called renal columns extend into the medulla and separate adjacent renal pyramids. The columns have a distinctly granular texture similar to that of the cortex. A kidney lobe consists of a renal pyramid, the overlying area of renal cortex, and the adjacent tissues of the renal columns. Urine is produced in the kidney lobes. Ducts within each renal papilla discharge urine into a cup-shaped drain called a minor calyx. 4 or 5 minor calyces merge to form a major calyx, and 2 or 3 major calyces combine to form the renal pelvis, which is a large funnel-shaped chamber. The renal pelvis fills most of the renal sinus and is connected to the ureter, which drains the kidney. Urine production begins in microscopic tubular structures called nephrons in the cortex of each kidney lobe. Each kidney has about 1.25 million nephrons with a combined length of about 145 kilometers. The kidneys receive about 20 to 25% of the total cardiac output. In normal, healthy people, about 1,200 milliliters of blood flow through the kidneys each minute, a phenomenal amount of blood for organs with a combined weight of less than 300 grams or so. Each kidney receives blood through a renal artery. This vessel originates along the lateral surface of the abdominal aorta near the level of the superior mesenteric artery. As it enters the renal sinus, the renal artery provides blood to the segmental arteries. Segmental arteries further divide into a series of interlobular arteries. These arteries radiate outward towards the renal columns between the renal pyramids. The interlobular arteries supply blood to the arcuate arteries, which arch along the boundary between the cortex and the medulla of the kidney. Each arcuate artery gives rise to a number of cortical radial arteries, also called the interlobular arteries. They supply the cortical positions of the adjacent kidney lobes. Branching from each cortical radiate artery are numerous afferent arterioles. These vessels deliver blood to the capillaries, supplying individual nephrons. After passing through the capillaries of the nephrons, blood enters a network of venules and small veins that converge on the cortical radial veins, also called the interlobular veins. The cortical radial veins deliver blood to the arcuate veins. These veins in turn empty into the interlobular veins, which drain directly to the renal vein. There are no segmental veins. Renal nerves innervate the kidneys and the ureters. Most of the nerve fibers involved are sympathetic postganglionic fibers from the celiac plexus and the inferior splenic nerves. A renal nerve enters each kidney at the ileum and follows the branches of the renal artery to reach individual nephrons. The sympathetic innervation, 1, adjusts rates of urine formation by changing blood flow and blood pressure at the nephron, and 2, stimulates the release of renin, which ultimately restricts water and salt loss in the urine by stimulating reabsorption by the nephron. When a substance is reabsorbed, it is reclaimed and then eventually re-entering the blood. So recall that kidneys remove waste from the blood and help to regulate blood volume and blood pressure, blood ion levels, and blood pH. In the kidneys, the functional units, or the smallest structures that can carry out the functions of the system, are the nephrons. Each nephron consists of a renal corpuscle and a renal tubule. The renal corpuscle is a spherical structure consisting of the glomerular, or the Bowman's capsule, which is a cup-shaped chamber approximately 200 micrometers in diameter, and a capillary network known as the glomerulus. The renal tubule begins at the renal corpuscle. It's a long tubular passageway that may be about 50 millimeters in length. Blood arrives at the renal corpuscle by way of an afferent arteriole. This arteriole delivers blood to the glomerulus, which consists of about 50 intertwined capillaries. The glomerulus projects into the glomerular capsule much as the heart projects into the pericardial cavity. Blood leaves the glomerulus in an efferent arteriole. It flows into a network of capillaries called the peritubular capillaries, which surround the renal tube. These capillaries in turn drain into small venules that return blood to the venous system. The process of filtration takes place in the renal corpuscle. In this process, blood pressure forces water and dissolved solutes out of the glomerular capillaries and into a chamber, or the capsular space, that is continuous with the lumen of the renal tubule. Filtration produces an essentially protein-free solution known as a filtrate that is otherwise similar to blood plasma. From the renal corpuscle, filtrate enters the renal tubule. The renal tubule has 3 crucial functions. 1, absorbing all of the useful organic nutrients in the filtrate. 2, reabsorbing more than 90% of the water in the filtrate. And 3, secreting into the tubule lumen any waste that did not pass and filtrate at the glomerulus. The renal tubule has 2 convoluted, which means coiled or twisted segments, the proximal convoluted tubule, or the PCT, and the distal convoluted tubule, or the DCT. They are separated by a simple U-shaped tube, or the nephron tube, also called the loop of Henle. The convoluted segments lie in the cortex of the kidney, and the nephron loop dips at least partially into the medulla. The regions of the nephron may vary by structure and function. As the filtrate travels along the renal tubule, it is now called tubular fluid, and it gradually changes in composition. The changes that take place and the characteristics of urine that result are due to the activities underway in each segment of the nephron. The nephron empties into the collecting system, which is a series of tubes that carry tubular fluid away from the nephron. Collecting ducts receive this fluid from many nephrons. Each collecting duct begins in the cortex and descends into the medulla, carrying fluid into a papillary duct that drains into the minor calyx. Nephrons from different locations within a kidney differ slightly in structure. Approximately 85% of all nephrons are cortical nephrons, located almost entirely within the superficial cortex of the kidney. In a cortical nephron, the nephron loop is relatively short, and the efferent arteriole delivers blood to a network of peritubular capillaries, which surround the entire renal tubule. These capillaries drain into small venules that carry blood into the cortical radiate veins. The remaining 15% of nephrons, termed the juxtamedullary nephrons, have long nephron loops that extend deep into the medulla. In the juxtamedullary nephrons, the peritubular capillaries are connected to the vasa recta, which are long, straight capillaries that parallel the nephron loop. Cortical nephrons do most of the reabsorption and secretion in the kidneys because they are more numerous than juxtamedullary nephrons. However, as you'll see later in the module, it is the juxtamedullary nephrons that enable the kidneys to produce concentrated urine. Next, we're going to take a look at the structure of each segment of a representative neuron. Each renal corpuscle is 150 to 200 microns in diameter. It includes both the glomerular capsule and the capillary network known as the glomerulus. The glomerular capsule is connected to the initial segment of the renal tubule and forms the outer wall of the renal corpuscle. It encapsulates the glomerular capillaries. The outer wall of the capsule is lined by a simple squamous capsular epithelium. This layer is continual with the visceral epithelium, which covers the glomerular capillaries. The capsular space separates the capsular and visceral epithelia. The 2 epithelial layers are continuous where the glomerular capillaries are connected to the efferent arteriole and the efferent arteriole. The visceral epithelium consists of large cells with complex processes, or feet, that wrap around the specialized dense layers of the glomerular capillaries. These unusual cells are called podocytes. Their feet are known as pedicles. Material passing out of the blood at the glomerulus must be small enough to pass the narrow gaps, called the filtration slits, between adjacent pedicles. Mesangial cells are specialized supporting cells that lie between adjacent capillaries. Actin-like filaments in these cells enable them to contract. In this way, these cells control capillary diameter and the rate of capillary blood flow. Several substances, including angiotensin II, ADH or vasopressin, and histamine, affect mesangial cell contraction. Some evidence suggests that these cells also make renin. The glomerular capillaries are fenestrated capillaries. That is, their endothelium contains large diameter pores. And the dense layers differ from that found in the basement membrane of other capillary networks in that many encircle more than one capillary. Together, the fenestrated endothelium, the dense layer, and the filtration slits form the filtration membrane. During filtration, blood pressure forces water and small solutes across this membrane and into the capsular space. The large solutes, especially plasma proteins, do not pass through. Filtration by the renal corpuscle is both effective and passive, but it has one major drawback. In addition to metabolic waste and excess ions, useful substances such as glucose, free fatty acids, amino acids, vitamins, and other solutes also enter the capsular space. These substances are reabsorbed before the filtrate leaves the kidneys. Most of this reabsorption takes place in the proximal convoluted tubules. Recall that the renal tubule consists of 3 segments: the proximal convoluted tubule, the nephron loop, and the distal convoluted tubule. The PCT is the first segment. Its entrance lies almost directly opposite the point where the afferent and efferent arterioles connect to the glomerulus. The lining of the PCT is a simple cuboidal epithelium whose apical surfaces have microvilli. You can check out table 26-1 for a little help on that one. The tubular cells reabsorb organic nutrients, ions, water, and plasma proteins, if they're present, from the tubular fluid and release them into the peritubular fluid, the interstitial fluid surrounding the renal tubule. The reabsorbed substances in the peritubular fluid eventually re-enter the blood. Reabsorption is the primary function of the PCT, but the epithelial cells can also secrete substances into the lumen of the renal tubule. The PCT makes an acute bend that turns the renal tubule towards the renal medulla. This turn leads to the nephron loop, or the loop of Henle. We divide the nephron loop into a descending limb and an ascending limb. Fluid in the descending limb follows towards the renal pelvis. Fluid in the ascending limb flows towards the renal cortex. Each limb contains a thick segment and a thin segment. The terms thick and thin refer to the height of the epithelium and not to the diameter of the lumen itself. Thick segments have a cuboidal epithelium. Thick segments are aligned with a squamous epithelium. Again, you can check 26-1. The entire descending limb is permeable to water. Its thick descending segment has functions that are similar to those of the PCT. It reabsorbs sodium and chloride ions out of the tubular fluid. The ascending limb is impermeable to water, but passively and actively removes sodium and chloride ions from the tubular fluid. Its thin ascending segment is permeable to sodium ions, which diffuse into the surrounding peritubular fluid. The thick ascending segment actively transports sodium and chloride ions out of the tubular fluid. The effect of this movement is the most noticeable in the renal medulla, where the long ascending limbs of the juxtamedullary nephrons create unusually high solute concentrations in the peritubular fluid. The descending limb is freely permeable to water, but not to solutes such as sodium and chloride ions. Water moves out of the descending limb, helping to increase the solute concentration of the tubular fluid. The thick ascending limb of the nephron loop ends where it forms a sharp angle near the renal corpuscle. The distal convoluted tubule, or the DCT, which is the third segment of the renal tubule, begins there. The initial segment of the DCT passes between the afferent and the efferent arterioles. In sectional view, the DCT differs from the PCT in that the DCT has a smaller diameter and its epithelial cells lack microvilli. Again, you can check table 26-1. The DCT is an important site for three vital processes. 1. The active secretion of ions, acids, drugs, and toxins into the tubule. 2. The selective reabsorption of sodium ions and calcium ions from tubular fluid. And 3. The selective reabsorption of water, which assists in concentrating the tubular fluid. The epithelial cells of the DCT near the renal corpuscle are taller than those elsewhere along the DCT, and their nuclei are clustered together. This region is called the macula densa. The cells of the macula densa are closely associated with unusual smooth muscle fibers in the walls of the afferent arteriole. These fibers are known as juxtaglomerular cells. Together, the macula densa and the juxtaglomerular cells form the juxtaglomerular complex, or the JGC, which is an endocrine structure that secretes the hormone erythropoietin and the enzyme renin. The distal convoluted tubule, which is the last segment of the nephron, opens into the collecting system. Individual nephrons drain into a nearby collecting duct. Several collecting ducts then converge into a larger papillary duct, which in turn empties into a minor calyx. The epithelium lining the papillary duct is typically columnar. Two main types of cells are found in the collecting duct: intercalated cells and principal cells. Intercalated cells are cuboidal cells with microvilli. Alpha intercalated cells and beta intercalated cells make up the population of the intercalated cells. Alpha intercalated cells secrete hydrogen ions and reabsorb bicarbonate ions, while beta intercalated cells secrete bicarbonate and reabsorb hydrogen ions. Principal cells are cuboidal cells that reabsorb water and secrete potassium. Together, these cells regulate the acid-base balance in the blood. The collecting system does more than simply transport tubular fluid from the nephrons to the renal pelvis. It also adjusts the fluid's composition and determines the final osmotic concentration and the volume of urine, which is the final product. We'll consider these activities of the collecting system later in the module.